Okay. So when you're, so I'm going to go into more of the technical part of how you do the sequencing in the Illumina platform. And there are 4 major steps that is involved in this. The first is the library preparation, cluster generation, sequencing, and then the data analysis. So in this workflow, there are many different steps that are involved in this. And in this presentation, I will be talking more on the different steps that are involved and also the different definitions or the words that are involved when you're talking about sequencing. So I will talk a little bit on how do you prepare the samples for sequencing, what are indexes, how do you optimize the library concentration, what are actually called reads, what is a flow cell, single and paired in sequencing, what is actually called as a cluster generation, and how this can affect our sequences. And coming to the sequencing, which is an Illumina signature, which is the sequencing by synthesis, then sequence generation in the MySeq specifically, and how is the sequence data stored. So I will talk a little bit on data analysis, but more of it will be continued by my colleague, which will be mostly on the quality of your reads, how do you take the sequences out of base space, and what you can do with the data. So coming to the first part of the library preparation itself, so you, as Mark was telling, that you can have cultures or strains or metagenomes like fecal samples, or you can have both as your start material. And when you're going to do the sequencing first, you need to work with the DNA. So you need to do the DNA extraction. So for the DNA extraction, you can use many of the commercial DNA extraction kits that are available, or you can use the native extraction methods. So once you have the DNA extracted, you can, in Illumina, it's kind of like less is more. So you actually start with around 0.2 nanogram per microliter of your DNA, which is of pretty good quality. And this will be your starting material to do the sequencing. So in the starting, so when you have your 0.2 nanogram of your DNA, the first thing that you do is fragment the DNA into an acceptable fragment size, which is around 400 to 600 base pairs. And this fragmentation can be done by different methods. So Illumina recommends the enzymatic fragmentation method, with using their in-house enzymes. But you can also use different sonicators for the DNA fragmentation. So once you have the DNA fragmented, so you have all these DNA fragments that is floating in your sample, but how will the Illumina, how will the sequencer know that these are the fragments that belong to this particular sample, and that how do you actually connect these DNA fragments to your sequencer? Which is why Illumina came up with these adapter sequences. So adapter sequences consist of 3 different parts. It has a sequence binding site, and this is a site which is similar for all of the fragments that will be there in your library. And you have another one which is called as unique indexes. So these are around 6 base pairs long, and they help the sequencer to identify these fragments belong to this particular sample. Because you may have around, I don't know, 40 or 50 samples in your library pool. But how will you identify that these particular reads belong to this particular sample? Which is why you use these unique indexes, which will be unique for your own samples. So once the sequencing starts, it identifies these unique indexes and bins them into a particular sample. So the last one that is there in the adapter sequence is the sequencing primer binding site. And this one is also similar to all the DNA fragments that is present in your library pool. And these primer binding sites are specific to your flow cell that is present in your sequencer. And I will talk a little bit more about this in my coming slides. So these adapter sequences are attached to your DNA fragments using a reduced cycle PCR amplification. So if you see those, can I do this? Yeah. So if you see those DNA fragments, so this is your DNA fragment, and these are your adapter sequences that are attached. So your indexes are in the middle. You have your sequence binding site on the side. And even if you've done this enzymatic or a sonicator using fragmentation, do you notice that some of the fragments are long and short? And so there is kind of an ununiform way of the fragmentation itself. So that will be in the next step. So this particular step takes around 45 minutes for 8 samples, which is actually pretty quick, you know. So now I was talking to you about the fragment lengths. And you don't want to sequence all your indexes and not have any fragments or any information about your DNA. So you want to have a normalized or a normalized DNA length so that you have all of your information inside these, between the indexes. So for that, you first clean your samples so that you specifically only choose fragments that are 400 to 600 base pairs. And for this, there was a new, I think it was in 2000, where they introduced or discovered these Ampere beads. And these beads can help you to selectively select only the fragments of particular fragment length. So if you're adding a 1:1, or the more beads you add, the lesser or the small fragments are selected. So you can adjust the concentration of the Ampere so that you select only the long fragments. So so using the Ampere beads, you clean up your PCR product so that you have a normalized DNA fragment length. And now you have all of these in different tubes, right? Because you have different samples, of course. And if you see the concentrations here, which is the concentration before, the concentration after your cleanup, you see that you have different concentrations of your DNA, or your library, sorry. And if you just use this, if you just pool your samples together and then you sequence it, the one that has the highest concentration will take up all your sequences, and you will not get anything from the rest because it's not normalized. So because of this, you normalize your library pool so that every library is given the same amount of DNA fragments. So you have this as your normalized library pools, and then you pool them all together into a normalized library pool that has all your libraries. So if you remember, I was telling you about the unique indexes. So you are pooling everything together, but then the sequencer can read these indexes and know that, okay, this library belongs to this sample, and it can differentiate. Okay, so from here, you do some more cleanups, and then you sequence it. But then all of this, you're doing it in really a tube. You really don't know what you're doing actually inside the tube. So you need to do some quality control measures, and I think it's very necessary. So this is very critical if you want to get a high output sequencing success. So when you want to do these quality control measures, you can do it in different steps. You can first check that if the input material that you have is actually of good quality. That can help a great deal when you're doing your sequencing at the end. Once you're doing the fragmentation of your DNA, and then you do the Ampere cleanup, you can again check if the size distribution, if you've actually selected the correct size for your DNA fragments. You don't want to have very short fragments that you don't get any information about, or you don't want very long fragments that it doesn't sequence completely. So you have gaps in when you do the assemblies. So these type of quality controls can be done using a bioanalyzer or quantitative PCR. So we have now talked about the technical thing of how do you do all these Illumina sequence prep, but then how will the sequencer know that these are the indexes that you have used? There are many indexes that are there for Illumina. So you prepare a sample sheet. So this sample sheet is kind of like, it's a code for Illumina to say that, okay, these are the indexes that I've added, look out for them. And this can be done using base space. And here I'm just giving you a screenshot of how do you make a sample sheet. So if you where is my, oh yeah, here it is. So if you just import a sample sheet that has your samples and the project name, for instance, all of your samples will get added into this base space, into this into your sample sheet. And this is where you say these are your indexes that you're adding, and then you can drag and drop and put that, okay, this particular sample A has index this one. I would recommend that even before you start your sequencing, you should have the sample sheet ready so that you know that this particular sample will have these particular indexes. And this will help you when you're doing when you're preparing your sequences, that you don't get confused and put some other indexes and you may get completely different results. Since you have a MySeq here, you can also do the sample sheet using Excel, where this is kind of the format that Illumina provides you with, and then you just add your sample IDs, your name, the indexes that you're using, and save it as a tab delimited format. And this, you can add it to the MySeq, and it will process your samples along with this sample sheet. Okay, so what do we do after we have all of these things ready? We sequence. So this is the sequencing in the MySeq. So the MySeq, you can have two types of kits. You can have the 75 base pairs one, which is 2 into 75. So if you have a paired end, you will have 150 base pairs. Or you can have the 2 into 300 base pairs, which is 300 and 300, which becomes 600 base pairs as a paired end read. This is actually the longest that Illumina provides. All other series are only until 150 base pairs. So that's 150, 150, which is 300 base pairs. And you see also the time that takes for 300 base pairs is also quite a lot. It's around 2.5 days. But the data that you get is also pretty good. So you can also do single reads where you can only have one end of the sequencing is, one part of the read is sequenced and not paired end sequencing, but you get much more data when you do paired end sequencing. And if you have around 10 bacterial genomes that you're doing a sequencing of 2 into 300 base pairs, you will have a coverage of around 75 to 300 times coverage, which means that a particular region in the genome will be sequenced 75 times or 300 times. So if you have 10 bacterial genomes, you will have 75 times the same. You'll have 75 times the same sequences of that particular region. So you can add more samples, is what I'm saying. That this is quite a lot. So you can add more samples. So now you're going to, here it is. So now it's the cluster generation part. So this is your cartridge where you load your library pool. This is your reagent kit, or where you have the reagents that are involved for the sequencing. And this small little thing that is here is called as a flow cell. It's that small, really. So when you have when you zoom into this part of the flow cell, you have this lawn of these sequencing primer binding sites. So this is the site that you see in the adapter sequence at the end. So once you load your sample inside, these particular your adapter, your DNA fragmented, which is has the adapter sequences, will bind to this site here. And once it binds here, there's a polymerase that will then extend its part to make it double-stranded. And then, so the way Illumina works is that it has two different sequencing binding primer sites. So one is called as A and B. And so what it does is it's called as a bridge amplification. So this part of the sequencing binder site will then bend. So one part of it will get cleaved away, and this part will again bend to form clusters like this. So these are called as clusters. So this is called as a cluster generation, where it's trying to find out that how many of the fragments will actually bind to the flow cell and how many of them can form this particular cluster. It's only after this particular clusters you will get a number that says, okay, you have this many clusters, like 300,000 clusters, and that means it will determine what will be your output. So these are the clusters that will be sequenced. So until now, there is no sequencing. It is just binding to this flow cell and making this bridge amplification and making clusters. That's it. So this cluster generation is very important for the sequencing. It is something from Illumina that they've said that if you have only too little of your libraries, you will get these type of sparse clusters. And when the sequencing happens, you will not get that much of output because there's nothing for the camera to read. This is kind of like a normal. So when there is a camera that is there, and then it sees the clusters, and then it sees, okay, I can spot the clusters. There seems to be a gap between the clusters. I can see clear definitions, and that will give you good quality reads. It could also be that you have too many clusters, that the whole flow cell is just covered, and it's not able to detect where is actually a cluster. It seems like everything is just into one big lump. So these things can help or influence the quality of your reads. This is how you may get like deletions, or that even the run could stop because it's not able to detect these clusters. So now coming to the sequencing part itself. So as I said, this is the sequencing binding primer site. And here you have once the sequencing starts, it will release one nucleotide to these clusters. Okay, these are all the clusters here. And this nucleotide is attached to a fluorescent tag. It is fluorescently tagged. And it also has this reversible terminator that does not allow this particular nucleotide to go and attach anywhere else. So this tagged nucleotide, if it is a match towards the read that you have, it will then emit this fluorescence. And this fluorescence is then recorded by a camera. So this is how the sequencing goes, and this is the cycle 6 of sequencing. And with each nucleotide that is released, it will detect these the camera will detect the fluorescence from these clusters. So the real version of all of this looks like this. I'm sorry, it seems to be a little bit blurred, but so this is the whole clusters, and it detects where does the fluorescence come, and it captures that as one particular picture. All these pictures are stored and then pre-processed after the sequencing to then detect the nucleotides. So what you see here is what you see inside a flow cell. But what do you see when actually the run is going on is again from base space, where you get this very interactive window where you can see how your run is going, how many reads have passed through a good quality, where if you see the Q score, where the Q score is like a quality score of the prediction that this particular nucleotide is actually the correct one. So if you, again, where's my, yeah, here. So if you see here, it shows this is something around 30, 40, and 50 Q score, and that means that it's of generally a good, it's generally of good standard. And if it says that, it says here around 80% of your reads that have been sequenced is of good quality. So this can determine that if the sequences or the reads, or sorry, if the sequences that are generated is actually of good quality. And this will be your first quality control measure. So here you can see that okay, there are this many clusters that has been formed, and these clusters are being sequenced. But how many clusters are actually good ones? How many clusters can you actually get some data out? And this graph can give you that information. So all of these information that it is kind of real time. So you can come anytime to the machine and try and look at this and see, okay, probably this run is going good, or that probably in the next run I may have to change something in it. And if you have base space, you can also view this in your own computer. So I've been talking a lot about base space, and there's going to be more information on base space in the coming talks. So base space is kind of Illumina's cloud service that is directly connected to your machine, if you have internet connection, of course. So all the data that you have on MySic, all the sequence data, all the quality control data, all the pictures of the clusters, everything will be stored in base space. So you can come anytime, just if you have everything stored on base space, you can come anytime, just open up and see, okay, I did this run and I saw that, okay, this is the clusters that I have. Or if you want to give this to an Illumina expert and say that, okay, this is how my run went, you can just share these files to the Illumina experts. So I think I'm done with my talk, and I think I've been very fast. But I have a video on what I've just been talking about, which probably I can play it to you, and you can understand all that I've been saying.
Well, how long is the video?
6 minutes.
Can I suggest that because it's